So hello everybody. I will introduce you Professor Luc De Meester, who is a Belgian and he holds a PhD in biology at Ghent University, at the city of Ghent, which by the way is a very nice, beautiful city. In early 2020, Luc became the new scientific director of the Leibniz Institute for Freshwater Ecology and Inland Fisheries, IGB, in Berlin. Full professor at Freie Universität Berlin and member of the Berlin-Brandenburg Institute of Biodiversity. He still is also partially connected with KU Leuven, the University of Leuven, where his research group there is still thriving with 4 postdocs, 8 PhD students, and 1 technician. But he is also building up a research group at IGB. Well, Luc has published several hundreds of papers with very broad contributions to many aspects of ecology and evolution. And by my last account, his papers have accumulated about 30,000 citations. So I can only say that his research interests are broad, but more recently have emphasized the interaction of eco-evolutionary processes and the environment along spatial gradients. His research involves metacommunity ecology, evolutionary ecology, stress ecology, and ecological genomics. And to address all these complexities, his group often uses a broad range of approaches, such as fieldwork, mesocosm, and lab experiments, resurrection ecology, genomics, and modeling. The overall aim of his research converges into understanding how both ecological and evolutionary response integrate in explaining how genotypes, populations, and communities deal with environmental challenges, including both natural, such as presence of predators, parasites, competitors, as well as human-induced stressors, such as climate change, pollution, and fragmentation. He is particularly motivated by eco-evolutionary feedbacks, and his recent research has provided important understanding on the role priority effects play on community assembly and on how crucial eco-evolutionary dynamics are to more accurately predict biological response to environmental change. And as anyone can assume by looking at his vibrant group, and as I can testify myself, Luc is also an excellent promoter. So Luc, thanks for accepting our invitation to share a bit of your work with us, and the screen is yours.
Andros, for this too kind introduction. It would have been great to meet you in person, of course, because it has been a few years since we met, and so that's a bit sad. And I want to apologize that if this would have been in a real meeting, I would have ended up in the wrong building. And but thanks to Andros, I actually found my way. Am I sharing now my screen? Does it work?
Yes.
Do you see the screen?
Yes, I see.
Yeah, okay, great. So I also want to thank Coca for a superbly organized virtual meeting. Coca, you and your team did a fantastic job. And it's of course a bit of pity that we cannot attend the meeting in real. And so to meet with friends and to have these inspiring brainstorms on moments when people would think that we are actually enjoying the sun and drinking a beer, but in practice we are actually making progress in science, I would say, by just having a good time and thinking out loud. And I also really would have enjoyed visiting Brazil. I've been there once and I had an amazing trip together with my oldest daughter to the Pantanal and the Amazon. And I mention this because for me, it is also really devastating when I noted that in the last year, the Pantanal was burning so heavily. And so that this amazing wetland was partly destroyed. And so I just wanted to mention that because it also, we often think about the Corona crisis, but it's sadly not the only crisis that we are facing in this world. So then Andros explained my new association. So I'm now affiliated with the IGB in Berlin and with the Freie Universität in Berlin, but still also with KU Leuven. My lecture slides will therefore sort of feature IGB, but also feature KU Leuven. And I have to say that, of course, I have been in the past 25 years connected to KU Leuven, and I should give them at this moment all the credits, because most of the work that I present here has been done at KU Leuven, of course, by the team that is there. My talk will have sort of 5 parts, and the first one is basically why Eco-Evolutionary Dynamics and what it is. And the second part is why should we take a landscape perspective. Then I will present some empirical studies on Eco-Evo in landscapes. Then I will present you the idea of evolution-mediated priority effects, because they are basically also leading to frozen signatures of Eco-Evo feedbacks in landscapes. And then some take-home messages. So the first part is on Eco-Evolution and Dynamics, so what and why. And yeah, so this is the way I look at it. So when we are studying natural and human-induced gradients in space or environmental changes during in the course of time, then we have ecologists looking at what that does to populations, communities, ecosystems, and we have evolutionary biologists who look at those same stressors and look how species respond to that in terms of their genes and their genetically determined trait values. And what Eco-Evo dynamics does is to bring those two things together, not just doing both ecology and evolution, but really looking at the interactions between those two. Because since 20 years, we sort of know now that species in many cases can evolve reasonably rapidly, which means at the same pace at which ecological processes occur, so that evolutionary changes can impact ecological processes. And so one way to look at this is that imagine that you would have a community inhabiting a pond that is exposed to climate change, nutrient enrichment, pollution, perhaps parasites, whatever. All these environmental changes are selection pressures. And they work on the traits among species to lead to species sorting. So the communities are adapting by changes in the relative abundance of species. But these same response traits and perhaps also interaction traits that determine the community structure, they may also evolve in each of the species of that community. In that population, these traits may also evolve. And if that genetic trait change occurs fast enough, so that the evolutionary trait change occurs fast enough, it may actually influence species sorting. So imagine the case where you would have climate change and all populations would perfectly track environmental change through time, then it's in principle possible that the community would actually not change, because everything in essence stays the same. All these species are tracking equally well and without any trade-offs climate change. I'm not suggesting that this happens, but it could happen in principle. And that would show that evolution can really impact species sorting and community composition. At the same time, of course, it also works the other way around. If species sorting is very fast, then local populations of a given species have no time perhaps to adapt. And if they have no time to adapt, then of course that species sorting will influence evolution. So it's really important to consider those two together. I want very briefly to illustrate this with not the idea of Eco-Evo, but the idea of rapid evolution by presenting 2 bits of our work on rapid or fine-scaled evolution to environmental stressors. And I want to use climate change and urbanization because I will also use them later on. So first, with respect to climate change, we did quite a bit of work there, but Aurora Heerts was the last in line to really focus on climate change. And she used an experimental setup that was performed by the group of Brian Moss, and he allowed us to inoculate all his great mesocosms with the same set of 150 clones, more or less, of a given population in the UK of Daphnia magna. And then half of the mesocosms were kept at ambient temperature, half of them were kept at +4 degrees Celsius. Aurora then, at the end of a 2-year selection period, she collected sediments and she hatched dormant eggs, and she quantified heat tolerance as the critical maximum temperature. The way we do that is sort of fun. You put the Daphnia in an Eppendorf tube, you put the Eppendorf tube in a thermocycler, and you increase the temperature of the water every minute, every 20 seconds with 1 degree Celsius. So what actually happens is that the animals always become more and more active because it's getting warmer, and at a given moment, they just faint. And the reason why they faint is that they don't have oxygen anymore in their muscles. So that is really quantifying a physiological maximum heat that they can tolerate. They are not dead. If you put them back in cold water, they become active again. So she did that for 10 clones from the ambient mesocosms and for plus 4 degrees Celsius mesocosms. And as you can see, the animals from the plus 4 degrees Celsius treatment have a higher heat tolerance than the animals from the ambient mesocosms. So in 2 years' time, this population was able to increase their heat tolerance significantly. What then Aurora also did is she took a sediment core from a lake nearby, actually, to that field station, and she hatched animals from the 60s and animals from the 2010. She quantified their heat tolerance, and again you see that the animals from the 2000s have a higher heat tolerance than the ones from the 60s. What this study actually shows is the first experiment actually shows that the current populations of Daphnia magna have the potential to genetically change and increase their heat tolerance, and this study actually shows that they have already done so. They did so in the past 40 years. The second gradient I want to look at is urbanization. That's the work of Christine Brans, who recently finished her PhD a year ago and is now doing a postdoc and in our lab. And she actually sampled a set of Daphnia magna populations along a rural to urban gradient in Belgium. So Flanders is very urbanized. And so she hatched animals from these different populations, and she then quantified heat tolerance again when they were cultured at 20 and 24 degrees Celsius. So the animals cultured at 24 degrees Celsius have a higher heat tolerance than the animals cultured at 20 degrees Celsius. This is not new, people knew that. However, what is new is that there is this beautiful gradient with along the rural to urban gradient. So the more urban the populations are, the more urban the environment is in the immediate surroundings of the ponds, the higher the heat tolerance of the animals. And so animals have genetically adapted to live in the city to have a higher heat tolerance. And if you look at the total variation in heat tolerance in that data set, you come to the conclusion that 40% of it is linked to that urbanization gradient, so to the genetic adaptation to the urbanization gradient. The other 60% is basically plasticity. So this is heat tolerance. Christine did many more studies, and she really found very cool results in the fact that, for instance, urban animals are smaller than rural ones, they produce more offspring, and they mature faster. So basically, urban animals have a faster life cycle than rural animals. And they can do so because they have a higher fat content, a higher sugar content, and a higher protein content than the rural ones. So they are, it's a total package. They are adapted to have a higher heat tolerance, they have more energy, and they can live faster. And these are just two examples of other studies. We looked at pollution, we looked at parasitism and predation. And in all of these studies, basically, we have evidence for fine-tuned spatial genetic differentiation or fast evolution in the order of a few months or a few years to those different stresses. And of course, you may think that Daphnia is a small critter, but this is a slide that I took from Andrew Henry. I adapted it a little bit because I added the water fleas and I added the rotifers to also pay tribute to the work that Steven does. This slide shows that, of course, we find this fast evolution. We have evidence for fast evolution in those small animals, aquatic as well as terrestrial, but also in mammals, in birds, and in fish. So there is more and more evidence accumulating that also vertebrates can show evolution across the time scale of, say, a few years. So if you have sufficiently rapid evolution, then you get into the potential that evolutionary trait change will impact ecology. And so this is a scheme of Eco-Evolutionary Dynamics put forward by Andrew Henry. And so basically it's saying that ecology influences the phenotypes of populations because of selection. And to the extent that the phenotypes have heritable variation, you can have evolution of those phenotypes. And when those phenotypes change, then you get into the Evo to Eco feedback loop, which is then that these phenotypes can change population, communities, and ecosystem processes. And what is really interesting is you have these two parts of it, but of course it goes much further, because basically this is an ongoing cycle. If you influence through evolution ecological settings, then these ecological settings can provide more selection pressures on the phenotypes, etc. And so that makes eco-evo dynamics so particularly interesting, because you can get totally different dynamics because you have this evolutionary trait change that is intervening there. I want to illustrate this Evo to Eco link with two studies, one of our own work with Daphnia and the other one on guppies. And so the first one is actually the impact of evolution on community level effects. And so that's work that Cathy Duvivier did the experiments and Jelena Pantel did the analysis and the writing of the paper. So here we wanted to go for the whole scheme, where we wanted to go both for the eco to evo as well as from the evo to eco cycle. So we first started with creating specific environmental conditions that would lead to evolution. Then we wanted to quantify how that evolution would impact community ecology. So what we did is we hatched animals from a specific population in Belgium. We exposed those animals to a selection experiment. And there we selected for specific conditions. I will explain them in a minute. We had these adapted populations, and then we used the adapted or the non-adapted populations to do a community assembly experiment. So first, the selection experiment. So this is basically in mesocosms where we created 4 conditions: presence, absence of fish, and presence, absence of macrophytes. We are in a shallow lakes conference, so I do not have to convince you that these are very important factors linked to selection environments in freshwater systems. And so we inoculated all these mesocosms with 115 genotypes, and we allowed them to, for 2 months, to adapt to those 4 ecologies. And then we isolated 20 clones from each of these mesocosms. And so we kept those clones for 1 year in the lab, and then the next year we did a community assembly experiment. So here we actually used either the adapted populations, so the ones, the clones that we isolated from the populations at the end of the selection experiment, or the original one, which is the non-adapted population. And so we allowed them to grow in the containers, and then added a pool of regional species, just sampled from the region, more than 2,000 individual container, more than 12 species. And after 1 month, we quantified community structure. So to repeat, we hatched animals from the field, put them in a selection experiment, used those to create adapted genotypes. And we did a community assembly experiment either with these adapted genotypes or with non-adapted ones directly hatched from the field. And the results are really striking. Jelena, the first thing she did was, of course, checking whether adaptation leads to higher abundances of Daphnia magna. And indeed, it is the case on average. If you look at the densities of Daphnia in the 4 different treatments, you have higher density in the case the Daphnia were adapted than when they were non-adapted. But the most intriguing question is, of course, whether this influenced the community structure of the non-Daphnia magna zooplankton. And we did a permanova on these communities of these different treatments, and we could show that fish and macrophytes have a highly significant effect on the community structure, which is no surprise to all of you. I think fish and macrophytes are strong determinants of community structure of zooplankton. But what is really striking is that the adaptation of Daphnia magna, so the adaptation of one species to these environmental gradients itself, also had a highly significant effect on community structure. And this is an NMDS plot where we have the 3 replicates of each treatment connected to each other. And so the blue triangles are actually the treatments with non-adapted Daphnia. The gray ones are with adapted Daphnia magna. So first of all, you see that there is quite high repeatability. So those replicates are quite close to each other. But the most important result is that you see a shift from, say, the blue to the gray triangles. The gray triangles are all very similar. And probably because the fact that the abundances of Daphnia magna were higher, Daphnia magna is a very strong competitor, resulted in a very similar selection pressures on the communities. An important result here is that if you look at the effect size and the direction of the effects for community composition of the ecological factors, fish and microphytes, and for the genetical factors, so the adaptation, then we see that it's actually the genetic effects are, first of all, the effect size is similar to the ecological factors, which is striking. I mean, we know how important fish and microphytes is, but still, adaptation to these factors is equally important, actually. But not only this, it's also in the opposite direction. So we see that actually evolution buffers for ecological change. And this is something that many people have found in the meantime, and that seems to be a reasonably general pattern in that, and often it is the case that evolution reduces the ecological pattern. And that's a very important observation, I think, that if you would expect a strong response to an ecological, an environmental change, that it's actually less strong than you would expect because of the fact that evolution may have tempered it. With respect to response at the ecosystem level, we actually did some experiments on that, but I would like to briefly mention this study of Bassar et al. Because it's quite unique study, I think, because it's in very near natural conditions that they worked. And so this is in small tropical streams, guppy populations, where it has been shown before that above the waterfalls, there is actually a different predator environment than below. There is more predators below than above the waterfall. And therefore you have actually a different selection for guppies. You have more colorful and bigger guppies above the waterfall than below. And so what Bassar et al. Did is they, in artificial streams, they created a number of conditions. And they had the conditions without guppies, and they had the conditions with the bigger guppies or with the smaller guppies, so the one above and below the waterfall. And they actually also inoculated them at different densities because they do occur at different densities. And so the circled situations are the densities and the identity as it is in nature. And so I cannot go through these results, it's too complicated. But these are important things, the total nitrogen flux, the algal stocks, the total phosphorus flux, so important ecosystem processes. And what you see for some of them is that the presence of guppies is very important. This is the situation, the absence of guppies, this is in the presence of guppies. So in these cases, you could say this is ecology. But here, for instance, the presence of guppies is actually less important than the identity of the guppies. And here you have a very complex situation that in the end you would not see the effect of evolution because at the right densities they have a similar effect, but here it's very different. So here again the presence is not as important as the identity of the guppies, and here you have a strong effect of both identity and densities. And so for every variable you have different patterns, but the point is that for several of them, it's actually much more important to know which guppy is there than to know that there is guppies, which is quite spectacular as a result, actually. Another reason or another link to ecosystems is, of course, that trait change, also evolutionary trait change, might impact tipping points. This was a study, rather an opinion piece led by Vasilis and Blake. And now there's a follow-up paper that is led by Catalina Chaparro, who modeled the effect of trait change and microphytes on tipping points in shallow lakes. And in future, I would, of course, very much like that we could also do this for Daphnia, for instance, with respect to the grazing capacity of in shallow lakes and ponds. So this was part one. So I hope to have convinced you that evolution can be fast and that therefore eco-evolutionary dynamics may be very important and actually do influence community and ecosystem processes. So why a landscape perspective? And I actually want to congratulate the organizers of the conference with a very beautiful logo for the conference. So that is also very useful now to show here. So why do we need a landscape perspective? So the point is basically that we are working with traits and responses to the environment. And so then space also becomes a very important factor. So you have a mosaic of environmental gradients in nature, and that is inhabited by metacommunities. but it's also inhabited by metapopulations. Imagine every single species here would also represent a metapopulation. So you would have gradients in the environment, presence, absence of fish, presence, absence of land, intensive land use, etc. You would have specific dispersal patterns, and all this connectedness, like Anna showed, and then the environmental gradients at both level of the population and the meta-community would be important. And that is basically the different components of this framework of the evolving metacommunities. So we have a metacommunity in a heterogeneous landscape that is actually in which the components can also evolve, and they may therefore influence the structure of the metacommunity. And of course, traits are very important there. So it's traits that, to some extent, at least to the extent that environmental drivers and species interactions are important, will drive this metacommunity composition, but it's also traits that determine metapopulation structure, and through evolution can change, influence metapopulation structure, and through that also metacommunity structure. So this evolving metacommunity framework is, in my opinion, a super attractive framework, but it's also a very complex one. It's not easy to get a handle on it. And to the extent, of course, I was mentioning traits, I was mentioning response traits and interaction traits, to the extent that also effect traits evolve, you would also have different impacts on ecosystem functioning, perhaps, like, for instance, algae control or primary productivity, etc. Another reason why space is important, or landscapes are important, is that there is actually already quite some evidence that evolution can modify spatial ecological patterns in space. And so this is a paper led by Mark Urban, who did an amazing amount of work screening literature, and there's 500 papers that were screened to see whether evolution would dampen or amplify existing ecological patterns, or even create new patterns. And so imagine the following: you have a productivity gradient, and that would result in a species diversity gradient. If, for instance, these species would be able to evolve so that they become more efficient with their food, they become physiologically more efficient, then they might actually occur across this gradient, and that would reduce the ecological pattern. You would not see an ecological pattern in diversity linked to productivity, but that would be a less strong pattern, or even the absence of a pattern. So that's evolution would dampen the pattern then. Evolution in some cases can also amplify the pattern. However, the basic message of this paper is that there is many cases in which evolution impacts the pattern, and in most cases it's dampening. It's an important thing to observe. I've mentioned it before. Evolution can buffer ecological patterns, and this is also here in space. It might be that in many cases we see much less strong ecological patterns thanks to the fact that species actually evolve. And another reason to include space is, of course, that space is a very important determinant in many ecological responses. Imagine the response to climate change, where you may have an interaction between local dynamics of adaptation in a local population, in a local community, but also the possibility of immigrants of pre-adapted genotypes and pre-adapted species. And in this context, for instance, we look in the first place, for instance, at these latitudinal gradients, but there's much more. For instance, like we have shown that Daphnia are also adapted to urban areas by being more higher heat tolerance. And to the extent that this heat tolerance is very important, you may expect that perhaps animals might spread from the cities into rural areas. And so this would create very different spatial patterning. I will come back to this race between adaptation and immigration, but once this race is important, space becomes very important. So I want to present now in part three empirical studies. The first one is on Eco-Evo along urbanization gradients. It's actually just mentioning of that study. This was a study that was coordinated by us on an urbanization gradient in Flanders. It was a quite complex sampling design with both urbanization gradients at what we call the plot level, at the larger spatial scales, but also at very small spatial scales within each plot. One of the outcomes of that study was that across different organism groups, there is a tendency for most groups at least to have smaller body sizes in urban areas. And also this holds for cladostrum communities. Andros did a very nice study on the metacommunity structure along urbanization gradients in this system. And one of the patterns, of course, that body size is strongly changing with the amount of urbanization. This is very similar to what Christine Brands found for the body size of Daphnia magna populations. They also become genetically smaller as you go into more urban areas. And I cannot go into detail because of time constraints and because it's a bit complex things to explain, but we actually quantified the contribution of intraspecific and interspecific variation in this gradient. And the end conclusion is that actually intraspecific variation was very important, but not the genetic adaptation in this case. In this case, it was actually rather ontogenetic variation. So the fact that animals start small and grow to larger sizes, that was the most important determinant of the body size distribution of total communities along urbanization gradients. The second study is, yeah, I show it. There is a part that is going to be published. There's a part that is not going to be published. It's perhaps also interesting for people because we had actually basically a pretty good idea, I think, but in the end, it was still difficult to make it real. And it's perhaps good for people to know that this happens everywhere. You try something and then in the end, it was less good than you thought. So what we actually try to do is to really look at those parallels in genetic composition, community composition, community trait composition, and species trait and individual species trait variation along environmental gradients. And so this goes back to this idea of Mark Velland that was also mentioned by both Pedro and Matthew, I think, in their talks about these parallels in the structuring process of communities and population genetic structure. So dispersal, gene flow, species sorting, natural selection, ecological drift, genetic drift, speciation, mutation. If these are so parallel, you could think, well, then there might be some really interesting interactions between, on the one hand, species composition and, for instance, genetic composition of individual species. And so that was basically the idea. And I can tell you this was actually a study that we started in 2006. And so it's very old, these data. And at this moment, they are still not published. And there is some specific reasons for it. But so what we did is we sampled 20 populations in Flanders. We sampled them from, we characterized the spatial settings, we characterized their environmental conditions quite in detail all. And so we selected orthogonal gradients of land use and fish predation and a little bit of parasitism also. And then we looked at communities, traits, and genomics. So basically, we did 4 parallel studies, you could say. We did a community composition, which is a metacommunity study. We did trait variation in the metacommunity. We did a common garden experiment looking at trait variation in the metapopulation of one species. And we then did a landscape genetic structure using neutral genetic markers. And our idea was, well, let's look at those patterns and then integrate them and come with a beautiful pattern of evolving metacommunities. And first of all, just to show you, this is Photoduct Behavior. This, you see that this is in the presence of fish, this is in the absence of fish chemicals. You see that the animals show actually more negative photoductic behavior in the presence of fish in all the fish populations and not in the non-fish populations. So there were some nice patterns of adaptive evolution to be found there. If we then did the first analysis, there was clear indications that community structure was much more explained by the environmental matrix and not by the spatial matrix. The same holds for trait variation in the communities. The same holds for trait variation within Daphnia magna. So here it's always environment rather than space who is a dominant factor. And for neutral genetic variation, this was not the case. There, space was more important. So this is not striking, but at least very nice to see that you have this environmental structuring for things that are linked to traits and not for things that are more neutral. However, the integration of it, because we wanted to know whether this matrix would actually influence the community composition matrix and so. And there we failed for the simple reason that we had 20 populations and our power was not strong enough. And so you should do this kind of things with 100 populations, but I can tell you doing it for 20 was an enormous amount of work. So I don't think it's so easy to convince anybody to do this for 100 populations. But then you would perhaps be able to have enough power to really understand the interrelationship between all those levels of organization, which was our original idea, but that original idea failed. Then Lien Govaert came into the picture, and we use this same data set now for a somewhat different analysis with some really interesting results. So Lien quantified the contribution of evolution of a single species, Daphnia magna. So these data, she used these data to quantify the contribution of evolution to community trait variation. So to look at how much does evolution influence community trait variation for 3 traits in those 20 populations. And I will not go into details, but in many cases, evolution has a very small impact, less than 10%. But in some cases, evolution has an impact more than 50%. So it depends very much on populations and on traits, what is happening. And that inspired us for another analysis on which we are very enthusiastic. Again, we are borderline with respect to power, but we are very enthusiastic about it, and we think this is something that people should do much more. So we quantify these evolutionary contributions to trait change, and then we have, of course, population genetic data from our neutral genetic markers, so we could look at what what the link is to this evolutionary trait change, or the importance of that evolutionary trait change to genetic factors, which is genetic diversity or population genetic distance of a given population compared to all the other ones, or ecological factors, because we have community data and we have community trait data. So we could also link it to species diversity or trait differences between Daphnia magna and the non-Daphnia magna community. So these 2 factors, if they would be the drivers for the evolutionary contribution to trait change, to community trait change, that would mean that it's actually evolutionary constraints that are determining the importance of eco-evo dynamics. Here we are talking about ecological drivers of eco-evolutionary feedbacks, the fact whether there is a high species diversity or the fact whether there is a lot of room to evolve because there is a strong trait difference between the focal species and the other ones. And you could, of course, also look at a purely environmental factor like the uniqueness of the habitat compared to the other ones in terms of environmental conditions. And through a Bayesian regression, we came to the conclusion that it is actually species diversity and trait difference between Daphnia magna and the non-Daphnia magna community that determine the amount that evolution impacts community trait variation. And that is really, really cool, I think. So what this actually means is that we have in eco-evo dynamics the idea that evolution impacts ecology. But here we show, or at least have very strong indications, that the degree to which evolution impacts ecology depends on ecology, depends on species diversity and on ecological opportunities. So how different the Daphnia magna traits are from the non-Daphnia magna community. The third empirical study is one that I'm also very enthusiastic about. It's very new. It's something that is led by Sophie Horvath and Xaba Vat. Sophie will give a talk tomorrow, another keynote. I'm very curious for that. And she and Xaba were enthusiastic to engage in this collaborative project. And I'm really grateful for that because it was a beautiful adventure, I think. So here we wanted to look at eco-evo dynamics in sodic bomb craters ponds in Hungary. And I should explain that a little bit. The sodic waters in the Pannonian Basin, it is basically a part of Austria, Serbia, and Hungary. So in Austria, there is a lot of soda pans. They are filled with sodic water. They dry up during part of the year. And then in Hungary, there is also soda pans, but there is also this beautiful bomb crater setting. This is bomb craters that were created in the Second World War, and they are really nicely isolated little patches in a landscape. And you see that they are very close to each other, but they're sort of a little bit bordered by a little dike, you could say, a little bit higher elevation, so they're pretty isolated. So we went there in 2018 and we collected not less than 400 Daphnia magna clones from different soda pans and bomb crater ponds, and we moved them all to Belgium and kept them in the lab. They are still in the lab. Then we did a common garden Ecotox experiment testing for salt tolerance of a number of these clones, something like 126. And then Xaba did also a live table experiment, and then Sophie and Xaba did a transplant experiment together with a number of people in Leuven. And so to just illustrate you how important Daphnia magna is in these sodic waters, so this is the sediments of a soda pan in Austria, and for those who know Daphnia a little bit, all these things here, they are all dormant eggs of Daphnia. So the sediments are largely composed of dormant eggs of Daphnia, so it's very easy to sample Daphnia. And I was very intrigued because you have these salinity gradients. I wanted to know with such a dominant species whether Daphnia magna evolution would impact the ecology of the systems. So the first thing we did is this Ecotox experiments. This is the average mortality along a gradient in the lab. So we put neonates at different salinity gradients and we quantified whether they survived or not. So this is the average mortality. This is for the bomb craters. This is for the soda pans. So you can conclude that the soda pans in general have a little bit higher salt tolerance than the bomb crater population. This is in line with the difference in salinity in the field. The soda pans have a higher salinity on, or can have a higher salinity than the bomb craters. So this is a regional adaptation, you could say. The two regions of a different salt tolerance. If you then look at the different populations separately, so this is the average mortality in the whole experiment, but now plotted against the salinity that we measured in the field. So this is for the soda pans and this is for the bomb craters. So the higher mortality means less tolerance. So you would expect that if there is local adaptation, that there would be a negative regression with this salinity gradient. For the soda pans, we didn't see anything. So soda pans populations have a higher salt tolerance than the bomb craters. However, there is no local adaptation to the different soda pans. And they are very shallow, they have many resting eggs, so probably there's a lot of dispersal amongst them. And so it's a rather regional selection that you get here. But in the bomb craters, even though they are so close to each other, they are pretty isolated. And here you see a nice pattern of local adaptation. The bomb craters have a higher salt tolerance in bomb craters that have a higher salinity. So we used this setting, the bomb craters, for our transplant experiment. So we did a field transplant experiment in 2019, where we had 5 ponds that we selected along the salinity gradient, and we did a full reciprocal transplant. We had the populations cultured in the lab under common garden conditions. Of all those 5 populations, we inoculated in mesocosms in all these 5 ponds. And so this experiment actually worked. And so we could monitor Daphnia densities and chlorophyll A concentration. And in this way, we wanted to look whether evolution to salt, to the salinity gradient would actually influence ecosystem features. And so these are very preliminary data. We did not move on too much because all of us were a bit too busy in the last year. So these are the 5 ponds, and we grouped them according to their mean EC50 for salt tolerance. And so this is Daphnia densities. And so this is actually the mesocosm experiment as was done in the low salinity ponds. So the lowest salinity pond. So this is the local population and these are the other ones. And you see that some of them have even a tendency for higher population densities than the local population. So there is no indication that local adaptation does do anything here for Daphnia densities. So if you would look at this pond, you would say Eco-Evo is not important. I mean, it doesn't really matter whether you're locally adapted or not. They are all doing more or less, they are equally successful. However, if we go to the other extreme, the highest salinity ponds, so then this is the local population, and these are the populations of the other 4 habitats which are less salt tolerant. And you see that here there is indeed a very strong effect of evolution. So only the populations that come from high salinity ponds do fine in this pond. So here we have a case where Eco-Evo actually does matter, but only in the high salinity pond. So only in the highly stressed pond. And we see a similar result for the chlorophyll A. So you would expect that chlorophyll A would be more suppressed if the Daphnia are doing better. And so we had a no Daphnia control. This is the red line here, and these other colored lines are all the different populations. And so in the low salinity ponds, you see a nice divide between the control, where you have no Daphnia, which have a higher chlorophyll A concentration than all the treatments with Daphnia. And the treatments with Daphnia do not really differ that much. So again, here we see that in the low salinity pond, it actually doesn't really matter whether the populations are locally adapted. The big difference is an ecological one. Is there Daphnia magna or not? So this is what should please us as ecologists, because we don't have to worry too much about the identity of our Daphnia magna. But when we go to the high salinity pond, the pattern is totally different and actually pretty beautiful. So the highest chlorophyll A concentration we still have in the absence of Daphnia magna, and the lowest concentration of chlorophyll A we have when we have the local Daphnia population inoculated in it. So the local Daphnia population is the one that can suppress the algae the best. The other Daphnia populations are intermediate. And here in this case, you see how important evolution is, because the difference here is not so much whether there is Daphnia in there or not. In some cases, it hardly makes a difference, but the effect is much stronger with respect to which Daphnia population is in the setting. So here we have a setting, low stress situation where ecology predominates. Here we have a setting where evolution actually is very important. So that leads us to the idea that there might be eco-evo hotspots linked to actually local stressful conditions. And I need to check a bit where I am time-wise, because I should not go too much over time. I finalized my lecture this morning, so I actually didn't know how much time it would take. So part 4 is on evolution-mediated priority effects or monopolization. Here I want to go a bit faster, because otherwise I will run into trouble. I mentioned this also because I'm still really intrigued by the idea, but also because there's a very strong spatial component also in this thing here. So I first want to explain what evolution-mediated priority effects are. So imagine an ecological world where you would have 2 species arriving in a single habitat with a certain time span in between them. So the 2 species have a different fitness, or if the 2 species have a different fitness, the first one has a lower fitness than the second one, then what would normally happen is that the second one will take over. So actually the color here, the more it matches the background, the better the species is adapted. So this one is better adapted than this one, and it will take over. We have no priority effect. However, if the 2 species are very similar with respect to fitness, it may be that the first species can already occupy a large part of the habitat, and that the other species cannot take over because they have an equal fitness. And in this way, it's only ecological drift that will determine what will happen. And in general, at least for a very long time, you would have a very strong priority effect where the first arrival species would dominate the system. My apologies. But that is, of course, the ecological world. Imagine that you would have evolution. And so in a world where species can evolve, the things can be a bit different, because this is exactly the same situation as here, the non-priority effects, with the first species being less well adapted than the second species. But here we have a certain time frame over which the first species can evolve and become better. And if during that evolution, it actually reaches the same fitness as the other one before the other one arrives, then you will have a very strong priority effect. So a setting in which you originally did not have a priority effect would suddenly become a setting in which you have a very strong priority effect, depending on the rate of adaptation and the time lag between the arrival of the 2 species. And here I want to stress that this is, whether or not this happens, is strongly dependent on the rate of adaptation of the resident population, and then of course the immigration of the other species. And that immigration rate is, of course, very much linked to connectivity, and that is space, in addition, of course, to dispersal capacity of the different species. So this is where space becomes a very important aspect also in this evolution-mediated priority. In a very extreme case, we actually know this evolution-mediated priority effects on oceanic islands in terms of adaptive radiation. There, the time lag between 2 species arriving is so long that you may have speciation, species adapting quite well to the environment and probably stopping further subsequent immigrants to arrive. And I cannot go into the details, but there's a beautiful study of Gillespie showing this pattern of adaptation versus immigration on the Hawaii Islands over a setting of about 4 million years. And then there is this experimental study of Ted Fukami, with bacteria, where he basically showed a very similar process, but at the time order of 4 days. And I really think this is beautiful as an idea that across those different time frames, you have actually a very similar process. So this led to the idea that you may have monopolization happening at different spatial setting and for different organisms, depending on, first of all, spatial settings determining how connected the system is, and so how many immigrants you will have. The second thing is that dispersal ability will determine how this translates into real immigrants. And the third element is, of course, the adaptation, the capacity of the different species to adapt. And so we made some predictions that for bird species, for instance, you may have this monopolization on very remote islands, but for smaller organisms with somewhat faster adaptation rates and in some cases not so strong dispersal compared to the adaptation, you may have already monopolization at the landscape level, different ponds in an area. And for bacteria, my prediction would be that it happens at the scale of your garden, basically. We model this is a model led by Joost van Overbeke, where we created landscapes with 4 species pre-adapted to 4 settings. And so if the world would be purely ecological, those 4 species would always occupy the right habitat in each patch. So we created empty patches or empty islands, and we let them through dispersal colonize, but our species could also evolve, and given enough time, for instance, the orange species could evolve to also occupy the yellow habitat. If you run that through time, you can have different patterns. This would be the pattern created by ecology. Every species colonizes the right habitat. This is actually patterns that are influenced by evolution. Here, for instance, the orange species colonizing the yellow habitat before the yellow species could arrive there, to the extreme that, for instance, the yellow species occupies a complete island before any other species could arrive there. And because the yellow species in the meantime is adapted to the habitat, it can monopolize it. To cut a long story short, the red line is basically the importance of ecology. The blue line is the importance of evolution, and linked to the amount of immigrants across among patches. And so basically, once you have less than 1 immigrant per generation, and especially if you have less than 1 immigrant every 10 generations, then you would see that evolution becomes more and more important. Some implications, and all these implications refer to patterns in space. So phylogeography, I cannot go into details, but for me, it's very striking that phylogeographic patterns are so persistent in nature. Over thousands of years, we still see the recolonization history of Europe for the different mammals species and other species here. Landscape genetic structure, where you in some species, notably Daphnia, you have much higher genetic isolation between neighboring habitats than you would expect based on dispersal rates. And we coined this isolation by colonization to explain it. An important aspect is that you would have a reduced match between species occurrences and the environment and metacommunities. So basically your environmental signal in metacommunity analysis would become weaker. And starting from that model that Joost developed that I showed a few slides ago, Matthew Leibold had this brilliant idea that we should also include disturbances in there, and that in this way we could actually show that, illustrate how this reduced match would increase due to the disturbances, and even to the extent that 2 species that originally are each adapted to 1 habitat patch. So you have the blue and the orange 2 environments in the landscape. We have 2 species that are nicely adapted to them. But over time, because they always need to recolonize patches, and because the species actually have the capacity to evolve and adapt to the other patch, you can create a situation that after, say, 10,000 generations, you would not, at the regional scale, be able to tell the difference between the 2 species. So they would both be adapted to both habitats, and they would have the same niche, basically, at the regional scale. However, at the local scale, they would always be very nicely adapted to the local patch. So that is a case where you can actually, through niche-based deterministic patterns linked, combined with disturbance, get regional neutrality. I'm also intrigued, but still did not find time to actually look into this a bit closer. But it's really interesting to look at the complex biogeography of species of especially unicellular species of protists, who often have very complex biogeography with both cosmopolitan and endemic lineages. So then I get to the take-home messages. So please, we should not ignore interspecific trait variation and evolution in our efforts to explain or predict response to environmental change. The effect sizes of genetic trait change or interspecific trait change are often as large as those of environmental change or even the presence of a species themselves. So if I want to get to a predictive ecology, for instance, in respect to climate change, these interspecific trait variation will be very important. Mind also that in many cases, evolution buffers for ecological effects. So the dynamics that lowers ecological scales or scales of biological organization buffer for patterns at higher scales. And so basically the take-home message is do not ignore evolution if you do not see ecological effects along a gradient or upon environmental change. So especially if you don't see strong patterns, it is potentially very important to look whether it's actually evolutionary trait change that balance for those patterns. Then the landscape approach is very important, especially because you may have these eco-evolutionary hotspots, and because in this concept of the race between adaptation and immigration, the connectivity link and the species richness link is actually very important. And so that leads to this, this suggestion to take trait-based and landscape-based ecology into account, and basically this evolving metacommunity framework to understand our world better. And as terms of future approaches, I would think traits, landscapes, eco-evodynamics. I have 2 times eco-evodynamics, that's a bit too much. Being very important to predictive ecology. And I'm super happy that Anna gave her talk just before mine, because I think honestly that networks of eco-evolutionary patterns and interactions is the way to go, where we would have networks of species composition and trait composition at both the intra and interspecific level, trying to connect those in landscapes. And I have no idea how to do that, but it would be super interesting to have brainstorms on these things. So with that, I want to end my talk. I want to thank the team in Leuven of postdocs and PhD students doing really great work and giving inspiring discussions, and also in the past and still going on, people that have been in our lab and are still involved in collaborative research. Thanks to all these people, and thank you for your attention. And for one strange reason, I deleted my acknowledgement slides where IGB was on, KU Leuven was on, and all the funding agencies. My apologies, this was accidentally dropped. Thanks very much.
So thank you very much, Professor Luc De Meester. I think it was a brilliant talk, right? I think it also shows a lot your enthusiasm and the vibrant research that your group is doing, because you have put several interesting results together. And I'm pretty sure most people here listening would like to do at least a whole 1 week course to be able to dig into more detail. And this is evolving so fast, right? So from the time I was in Leuven, that I could even do a new PhD on this topic. So it's very exciting. We have got a number of questions now, so I will go into them following the order that they have arrived, right? So the first is by Annette Janssen. She says, thank you very much for the nice talk, Luc. You said that the Daphnia were hatched for the 60s and current. I am not an expert in this field, but can the eggs survive so long? And might it be that the old eggs are of lower quality, thereby affecting the results between the data for now and the 60s?
Yeah, thanks very much, Annette, This is an important question. I'm happy you ask it because then I can explain. So first of all, indeed, Daphnia X can survive for several decades. There is at least one study that hatched animals of 600 years old. So this is truly spectacular. So it's very difficult to hatch them, but if you manage, that's of course brilliant. So indeed, it's possible to hatch animals from the 60s. And I think your question is a very logical one, whether those eggs would have a lower quality. The whole point is we hatch them and then we keep them in the lab. And Daphnia we can culture as clones. So we keep them in the lab for as many generations we want, say 5, 6. You give them good food. So the animals you work with are as young as any other animal. And so they come from an old egg, but you have kept them in the lab for several generations. So the quality itself might be a little bit lower, but that's just at the start of their hatching. So when you do experiments, it's totally different.
Okay, so moving on. Carlos Iglesias, thanks, Luke. I wonder when you talk about evolution, if you already have had the chance to identify genes mutations. And if so, in the case of Daphnia, having alternate life cycles, have you already identified if these mutations appear during parthenogenic or sexual reproduction phase?
Yes, thanks, Carlos. This is again a very good question, of course. And yes, we did. But I could not present that. There was some time limitation. It's not directly linked to landscape, although a little bit. I was contemplated to put it in there. But Anurag Chaturvedi did a full genome sequence analysis of our one of our studies. It's a resurrection ecology study. And actually the study I presented with the 20 populations in space, where we did the community and the traits and etc. We also did full genome sequences of those clones. So in total, that was about 500 clones. It was a very heavy investment to do it, but Anurag now has his data analysis ready. The paper is in, the first paper is in revision in Nature Communications at this moment. And so it's really intriguing. So first of all, we see a lot of genomic responses, but they are all small scale. So it's many genes evolving with small effects. It's not a very simple system. It's hundreds of genes that are evolving. And but what is really important, and that goes directly to your question, is that we could show that there is an enormous amount of genomic variation in Daphnia populations. So in the sense that any selection we had in that specific study could be supported by the genetic variation that was present at the start of our study. So there was no need for mutations. There was no need for novel mutations or immigrants in the population. Everything could be done based on standing genetic variation. And for me, the most spectacular results of that paper, and so it's hopefully going to come out soon, is that we did a rarefaction analysis. And so for all the genetic variants that you need to evolve for a certain selection person, in this case it was fish. So for all the genetic variants that you need to evolve, you actually only need 5 individuals from the landscape. So we actually screened 20 populations. So we could sample individual populations and see how the genetic variation would accumulate. And we could show that 5 individuals, only 5, would give you 80% of all the variation that you would need for this rapid evolution. So the genetic variation at the regional level is very high, and therefore, because there's a few immigrants from different populations in every habitat, you also have a high starting genetic variation at the local populations. So yes, there is genetic variance, but largely fueled by standing genetic variation that is present in the field, in the landscape, and in the local populations.
Right, the next question is by Marcus Herman, and he says, "Hi Luke, great and interesting talk, thanks. Do you think we can further translate the observed thermal adaptation of species as you showed in the first study for Daphnia magna in temperature increase of +4 degrees to explain possible latitudinal differences in species thermal sensitivity? Meaning species that always lived under warmer conditions could, based on their evolutionary background, be better prepared for future climate change, including rapid temperature change than species from colder geographic areas?
Yes, I think, again, thanks for the question. And yeah, you're probably right, of course. If you have experienced warmer environmental conditions, you could, in principle, probably have more genetic variation for to faster adapt to warming situations. I would even go a bit further, and this is purely speculative, but I actually deep down I believe it's true. That a lot of the genetic variation that you would see, for instance, in the Daphnia population in Belgium, has still the signature for the ice ages and interglacial periods in the past. I mean, many genes are actually 100,000 years old or something like that. So many of those those species and these genes actually survived several ice ages and changes in environmental conditions. And so it's possible that if you have this high genetic variation, that at very low frequencies, old variants remain in the genome that may actually allow adaptation to other conditions in the future. So I really think that what we get is always a sort of a mixture of regional variation, but some of this variation is pretty old. I think in many cases we will come to the conclusion that the genetic variants that we are dealing with are older than the ice age. So we are integrating a lot of genetic variation from the past, actually.
Okay, so the next question is from Sarah's Institute, which means it's by Nestor. So Luc, very good and inspiring lecture. Thanks a lot. How much conditions the rapid adaptive response in Daphnia depends on the particular characteristics of the Daphnia life cycle? Combination of forms of reproduction, parthenogenesis, resistance structures, etc. Some of these characteristics are not found in other species.
Yeah, Nestor, thanks very much for that question. It's of course a question that I get often, and it's a very logical question also. I have a number of answers to it. I have to say that, say, 15 years ago, I was very prudent saying, well, this is Daphnia, we have to be very prudent, etc., etc. Now, if you see that there is a lot of eco-evo-dynamics shown in guppies and stickleback and all these other organisms, then of course, it's a little bit longer time scales, but still there is the same eco-evo-dynamics. So I would like to make a few points. So first of all, you're very right. But second, this special form of reproduction of Daphnia is not rare. I mean, a number of insects have it, a lot of parasites have it, all the protists have it more or less. So they all mix sexual recombination with clonal reproduction. Many plants do it. So you have many organism groups that actually have this combined life cycle. So that's one thing. And the second thing that I would like to stress is that this life cycle doesn't make, first of all, our studies much easier. Let's emphasize that. It's much easier to work with Daphnia than, for instance, with copypods for such research. And it makes some of the aspects going faster. That's true. At the same time, I think the key to everything that I'm saying here is basically not so much a reproduction cycle, but the fact that Daphnia have large populations. So the fact that these large populations result in high genetic variation, high standing genetic variation that leads to rapid responses. I mean, there is many studies on insects not having these parthenogenetic life cycles that show this very rapid adaptation to Drosophila adapts very fast to certain conditions. I was involved in a beetle study showing very fast adaptation to conditions. And I think it's rather population sizes. So the big divide will be that you have certain organisms, because they are rare, because they're hunted, because they're large, that are smaller population sizes, less genetic variation, less standing genetic variation, more problems.
Okay, so next question is by Pedro Perez Neto. I will tell Coca that he might interrupt me later on if he thinks he should do so, but I will read this one by Pedro. So, hi, Luc. It would have been great to be in Brazil with you. Great talk as usual. the concept of priority effects in my mind is heavily driven by a target species A with greater fitness arriving before other lower fitness species, say B, C, and D, regardless of the order of the arrival of these later species. I would think that the order of the later arriving species should affect the overall fitness of all populations within a patch. Would you agree that this would be also important for predictive ecology? And if so, how do you consider these more complex domino effects of species arrivals?
Yeah, first of all, I fully agree, Pedro, and yes, indeed, it would have been great to be there. Yeah, I fully agree with you, Pedro. So this priority effects scheme that I propose is, of course, very simplistic. So I think the way to tackle this would be doing modeling with many species at the same time. And you and I, we know, of course, that we have actually engaged in trying to build such a model, and it would be really great if we could, in one way or another, keep this going. In parallel, I can sort of say that we have done a model with different clones within species, so it's not the same as among species, but within species. Lawrence Kilsdon, who did that, and in his modeling, it is really striking that if you keep on inoculating more and more species through time, that is still the landscape genetic structure is nearly totally determined by the first 20 arrival genotypes of a population. So you may have indeed this cumulative effect, but still it would perhaps be for the real first arrivals. Of course, all this does not matter if you have species that are clearly superior, and so then they take over, and then there is no priority effects, of course. I hope that answered your questions, at least more or less.
So Coca, can I go on? All right. So I think there is one by Ali Gare. I don't know the order now because it's a bit confusing, but thanks for a very interesting talk. How long do you think it takes for Daphnia to evolve heat tolerance? Is it 1 year or a few months also enough? Also, how do you think heat tolerance affects stoichiometric requirements?
Yeah, thanks, Ali. I think it can be fast, and we have actually data. I didn't show that, but Helena van Velke, PhD student in our lab, did an experiment where by accident she ran into a heat wave, and we used the data of that experiment to actually quantify the responses. And that heat wave was about 3 weeks long, and in 2 months she had a significant response to that heat wave. So, and that was again simply a population that we inoculated as a combination of clones. So yes, I think it can happen in one growing season. And I think if you have a heat wave, that you might have a signal of that in those populations. And I think the effect of that on stoichiometric requirements would be really, really interesting to study. We did not engage in that, and I do not immediately see papers that would give me an indication of an answer, but I think it's a really important thing to study, how that would affect these stoichiometric relationships. So a really very important suggestion, I think.
I think we have 3 more questions. So the next is by Libing Zul. Thanks, Luc, for the very interesting talk. How do you think that the evolution would affect ecosystem functioning or stability?
Yeah, thanks, Libin. Yeah, so more or less in line with what I suggested, I think evolution may, in some cases, have this stabilizing factor. In that model study that Catalina Chaparro is doing on these microphytes, there is actually 2 sides to it, because it has a stabilizing effect to prevent the regime shift in one direction, but it also has a stabilizing effect to prevent the regime shift in the other direction. So it may also make restoration efforts more difficult. So, but in general, my feeling would be in line with what Mark Urban's literature overview suggests, is that in most cases it will increase stability, and in some cases it will reduce it. And it depends basically what evolves. So some of the, if a predator evolves, whether a prey evolves, that may have different effects on the stability of an ecosystem.
Right, then there is another one by Esteban Ortiz, a really interesting talk. Luc, thank you very much. Selection of individuals can affect community structure. That is richness, relative abundance distributions, body size distributions, trait variation. But do you think it's possible that selection occur on different topological characteristics of, for example, food webs favoring the emergence of some specific topological patterns when communities are under different environmental or biotic stressors?
Yes, those are really intriguing ideas, I think, and I would say yes, I think so. I would not have specific predictions, and what would be a really interesting eco-evo twist to that is that selection might influence food webs, but then selection on the different members of the food web may change that response in the topology. So sure, absolutely necessary to study, but we are not there yet at this moment. I think I don't think we have very clear evidence for it.
Okay, and the final question by Evaldo Leitão. Thanks for the talk, very interesting. I was wondering, are there costs to adaptation? And another question, how about environments where there is no steady increase in environmental stress, but instead a higher variation? Would you expect a role of phenotypic plasticity would be predominant than adaptation?
Yes, thanks for the question. Yeah, important points, of course. I mean, first of all, there are costs to adaptation. I should also say that I'm always saying that they can adapt to this and they can adapt to that, but of course, we're only looking at populations that actually survive. So if you would have a stressor that nobody can survive, you would not find the population to actually test for the adaptation. So this is also an important point. But yes, and we have shown this, for instance, if you have adaptation to pesticide tolerance, we show that animals became more susceptible to parasites and stuff like that. So clearly there are costs of adaptation. There may also be a very indirect cost in the sense that selection may, for instance, reduce the capacity to respond to evolution in the future because it erodes genetic variation. And then, of course, your idea of the fact that you may have fluctuating environments and whether that would support more plasticity versus genetic variation. My attitude towards that is that you will always tend to have a combination of things because, I mean, yeah, nature is not geologically oriented. So the point is you will have selection for different genotypes, but they will also show phenotypic plasticity responses. You often have selection for phenotypic plasticity, by the way. And so which ones will be the most important for survival? That will depend on the speed by which the fluctuations occur. If they occur very rapidly, plasticity may be the only way that confers fitness. If they occur a bit slower, it might be useful, or it might just happen that part of it is through evolution. Yeah, so I think of combinations of these things, but in many cases, we see, of course, a lot of phenotypic plasticity in Daphnia, and I should absolutely emphasize that.
So thank you very much, Luc. We are just on time.